getting vaccinated and enjoying a nice spring. I'm going to go through some quick updates on the activities of the PRISM and what's going on, and including a few words about fundraising. And if anybody's got any questions about the PRISM itself, we can deal with that. And then I'm going to give some updates on what's happening with the spotted lanternfly, as well as with the verticillium wilt and how what its status is in terms of becoming a tool to deal with Ailanthus. And then I will hand it back to Beth to introduce Kelsey. So here we go. In terms of landowner activities, for 2021, we will have our 4 quarterly meetings, as we've had for in some number of years now. Our tabling events continue to be on hold until other people get their events going. Speaking engagements, well, we are making some interesting progress this year already, which is we did 10 speaking engagements in 2020, and we've done 8 so far this year. And there's more already scheduled. So if anybody's got an interest in having us come and do a speaking engagement for your group, we're happy to do that. In terms of workshops, we have our usual 9 workshops planned, 3 spring, 3 summer, 3 in the fall. But if time permits, we're hoping to do a 2.0 pilot, if you will, which would start talking about a whole nother round of plants that we don't normally talk about quite so much, and get into some interesting new topics. We're continuing to publish our quarterly newsletter, and of course our materials continue to be available on our website. So some other interesting statistics. We've had over 1,100 attendees at our events last year, and we've already had over 1,000 so far this year. And that doesn't count the fact that we've started co-sponsoring some other events where we've had almost 4,000 people so far show up at some of those other events. So our reach is expanding. And that's also evidenced by what's happening with our email list. We're now over 2,400 people. At the beginning of 2020, we were like 1,300 people. So we've added over 1,000 people in the last 15 months. So that's all great news. The PRISM is alive and well and continuing to progress. In terms of landowner financing, we're kind of working through our existing programs. We're now in the final year of our NRCS RCPP program, which provided funding for 94 landowners to treat over 2,000 acres. This is going to be year 4 of our DuPont settlement grant for treating wavy leaf grass at several sites. And this will be the 2nd year of work with our U.S. Forest Service grant to treat wavy leaf at some additional sites. So, things are progressing there. And we're constantly looking for other opportunities to acquire funds to do field work. Other key activities on a statewide basis, we are working to re-engage. The pandemic got in the way as well as the legislative session. So we'll be working with the secretariats that report to the governor, as well as VDOT, Wildlife Resources, DCR, DOF, VDACs, and perhaps go back and have another conversation with the First Lady. So some interesting things going on here, which is we are a party to, if not one of the primary drivers of a VDOT study that just kicked off in January. And so VDOT has hired Virginia Tech to go out and sample plants at hundreds of roadside locations throughout the state to inventory both the native plants growing there as well as the non-native plants, with the view toward making recommendations back to VDOT in terms of what should they be planting and what native plants should they be planting along the roadsides instead of predominantly non-native plants, including some invasives. So we're trying to kind of get VDOT's practices to be more native plant focused. And in addition, in this last legislative session, we were one of 5 organizations that came together to drive a bill through that's waiting the governor's signature. And this is a bill that asks VDACs and DCR to form a working group to come back and recommend options to stop the sale of invasives. Because if you look at the 90 plant list put out by DCR, somewhere around half of those are actually available in the nursery trade and the retail trade in Virginia, which is not a good thing. So we're working with that. Also, another thing we work on, of course, is nominations for the noxious weeds list. There were 6 of them that we succeeded in getting added officially last July. We actually have a meeting tomorrow where we'll be discussing the next round of nominations. Hopefully we'll get perhaps another 10 plants set up to go through the 2-year process of approvals that's required to actually get them on the list. So we're working on that one actively. And we have this project going on with UVA with Kelsey Huelsman, so I don't need to talk about that because she's going to talk about it a lot. And so we've got help available if any of you are out there wanting to start a PRISM to cover another part of the state. In terms of fundraising, just a reminder, the PRISM is 100% funded by grants and donations. The core of that is 2 matching grants, one from the Virginia Environmental Endowment and another from a family foundation. Each of those 2 grants requires us to find an additional $20,000 of matching money from people like you, which we're pleased to report. We tend to do pretty well with that. Q4 last year, we did the Family Foundation grant and found our matching money for that. We hope to have the next Virginia Environmental Endowment grant come through somewhere around now, could be any day now. And then we're going to be looking for money to match that one as well. So you may see something come through on that. And this is just a reminder that we are now a separate standalone corporation, 501c3. We're no longer operating as a project of the Shenandoah National Park Trust, which means donations can now be made out directly to the Blue Ridge PRISM or Blue Ridge PRISM Inc. So just a few reminders of things everybody can do to help with invasive plants in terms of both understanding what you have, helping your neighbors understand, record sightings of less common plants in one of the phone apps. You can nominate plants for the noxious weeds list. There's lots of volunteer things you can do. You can plant native plants. You can have us come speak to your group, or as I mentioned, help form a PRISM in another part of the state. And again, reminders, as Beth mentioned, we have our next round of workshops coming up. There's one on Wednesday, one on Saturday, one on Thursday. 2 are in the afternoon, one's in the morning. And of course, out on our website is lots of additional information, both around our events as well as how to make donations. You can send us questions, photos of plants if you want help identifying something, or if you can request a speaker for a group. So I don't know if there's any questions, Beth, out there around any of this. If not, I'll proceed. And Beth, are you there?
Yes, I am, Rod. Sorry, I couldn't find my mute button.
Yeah, no problem.
No questions have come in. Does anyone in the audience have a question? If not, we'll move on.
All right, so I've got a couple more topics to go through here.
Okay, well, go ahead, Rod. Thanks.
Yeah. So I'm going to talk a little bit about the spotted lanternfly. So this is a one-page alert document that we sent out. You can see the pretty picture in the upper left-hand corner. If you see this bug, you need to report it. This is a big deal. So here's one slide on this topic. So yeah, I characterize this as the next bug that we're all going to learn to hate. It's a really messy thing to have feeding on the trees in your yard, but it actually feeds on more than 70 plants. However, its clear favorite is Ailanthus. And right behind that is grape, so which means that it feeds on one of our nasty invasive trees, and it also is an agricultural pest and causes a lot of harm. I think they're they're projecting in Pennsylvania, by the time the state of Pennsylvania is covered, it's going to be costing $50 million a year. And most of that's probably going to be in the grape and vineyard industry. In Virginia, we have a quarantine area, which is currently Frederick County up with Winchester. But you know, the process is in motion, and right as we speak, to add Clark and Warren counties to the quarantine area, which means businesses in those areas need permits and training before they start shipping things out of those counties. So this is a big deal. And of course, one of the really key questions here is, how important is Ailanthus, specifically the Ailanthus plant, to the life cycle of the spotted lanternfly? Recently released research says that it's true that unfortunately the spotted lanternfly can live without Ailanthus. However, the good news is that it has 77% fewer egg masses because without Ailanthus, it matures more slowly. And so it has less time, you know, to produce egg masses, you know, before it gets hit with the frost later in the fall. So the big question that everybody's going to be asking is, should we take out our Ailanthus before the bug arrives? And based on this latest research, you know, all the experts that I talked to, now seem to all agree that if you have the wherewithal to take out your Ailanthus before the bug arrives, that would probably be a good idea. So that's just a heads up. So the other thing here is, as noted at the bottom, is there may be an opportunity, if anybody's interested, and people are still working this out, to put up traps, if you will, on your property on Ailanthus. You need to still have an Ailanthus tree that's more than 5 inches in diameter. But you know, people will be putting up traps to detect when the spotted lanternfly actually arrives in your area. So there may be an opportunity to participate in some of that as if you will, as a citizen scientist. So that's the spotted lanternfly topic. And then I've got one other Ailanthus topic. Many of you have been on our previous quarterly meetings, wherein we've had a report out of the status of the verticillium wilt as a biocontrol. This is being done by a combination of Penn State, the West Virginia University, and Virginia Tech. And so I traded notes the other day with Professor Salem down at Virginia Tech, inquiring as to where do we stand in terms of getting the verticillium wilt registered with the federal government as a bioherbicide. And his best guess is we still got about 3 years worth of work to be done to get that to happen. And this is time that's going to go into carefully controlled studies to make sure that this bug isn't going to inadvertently attack other things that we care about, as well as testing to make sure that the version of it that they're using in Pennsylvania also works in Virginia. Because of course, on the one hand, everybody is properly sensitive to not wanting to be spreading something around that's going to cause harm that we don't want. On the other hand, unlike a lot of other biocontrols, this is something that's already in the wild in Virginia and in Pennsylvania. So that hopefully that will help facilitate getting it approved. One last topic, which is truly tentative, is I've started asking the question as to whether there is any possibility whatsoever of getting an emergency use authorization for the verticillium wilt, so that we can sidestep some of the lengthy time required for the normal process. And I've actually been told that there is a process for doing this, and it's not impossible, although it's extremely difficult, and it would have to be driven not by us, but by the Commonwealth of Virginia. And this process would probably take at least 18 months. Nonetheless, if we can get something done in 1.5 years quicker by going through this route, it may well be worth it to a lot of people in Virginia. So that's those are my updates, if you will. I'm going to turn this back over to Beth, and she's going to introduce Kelsey Huelsman. But I'm going to take the opportunity to say I've been working with Kelsey. She must be close to 2 years at this point, on this project, and she's doing wonderful work. There's a ways to go to get it to where we can all use something like this. On the other hand, the results to date, I would say, are quite promising. So I'm enthusiastic, and I love seeing what she's doing here. So, Beth, I'm going to turn it back to you.
Okay, cool. Rod, we did have one question that came up.
Okay.
Is it about the verticillium wilt authorization? And the question is it the identical process for authorization of a chemical herbicide under emergency use?
Well, for the emergency use authorization, I believe that process, based on what little I've learned about it so far, I believe it is the same. And it's obviously tricky, and it's highly bureaucratic. I mean, in terms of there's lots of there's an application, and the application is lengthy with zillions of questions, and it's a lot of work to make sure you've really got things thoroughly documented, and you got it right. Yeah, because nobody wants to do it wrong. So it's a big deal, and it's complicated, but it can be done. So that's, and I'm pretty sure it's the same process. It's called Section 18, and I've seen a little bit of the paperwork that's required for it.
Thank you, Laura, for that question. And that's all the questions we have, and that was so. Rod, if we'll go ahead and turn the screen over to Kelsey. And Kelsey, if you'd like to go ahead and cue up your presentation, I'll introduce you. So Rod, thank you for the update and the updates on the verticillium and the spotted lanternfly.
My pleasure.
So I'm happy to introduce Kelsey Huelsman. Kelsey is a 5th-year graduate student in the Department of Environmental Science at the University of Virginia. Her current research at Blandy Experimental Farm in Northwestern Virginia is focused on remote sensing of plants with applications to aid both land managers and ecologists. Prior to beginning this project on the remote sensing of invasive plants, she earned a master's studying ecosystem functions in plants and soils, also at Blandy. In her spare time, Kelsey does work with Richmond Tenants Union in Richmond, where she lives, and United Campus Workers at UVA, where she works. She also enjoys growing plants, cooking plant-based meals, and playing tennis. Kelsey, thank you. I'll turn the screen over to you.
Thanks, Beth. Can you see my screen?
Yes, I can.
Okay, great. So as Rod and Beth both said, I'll be talking about the research that I've been working on at UVA, particularly collecting data up at Blandy Experimental Farm, which is in Northwestern Virginia in Boyce. And the data that we collect is via drone, and it's called hyperspectral data. And we're using that to try to identify and map invasive plant species. So the title of my research sounds kind of complicated, but I'm going to walk you through what matters and what it all means. So we'll start with what I know everyone is here for, which is invasive plants. And the 3 plants that I'm focused on at Blandy, trying to detect and map, are what is pictured on the far left, buckthorn, what is in the center, autumn olive, and what is on the right, tree of heaven, or ailanthus, which Rod was just talking about. So you obviously know tree of heaven, you probably know autumn olive, you might know buckthorn, but it is really quite prevalent at Blandy Experimental Farms. So that sort of is like an added bonus that I'm looking at, especially because there's so much buckthorn at Blandy, and it seems to kind of be impacting succession in forest development. So this is where buckthorn, this is a map actually for common buckthorn, which is closely related to Dahurian buckthorn. So you can see how widespread of an invasive plant it is. Autumn olive, even more widespread across Virginia, and tree of heaven, which we just talked about, is obviously in the running to be on the noxious weed list, so is important for us to be able to detect and map. So the work that I do at Blandy is drone-based, and these are some photos of me with the drone. There are drones that come in a lot of different sizes, and ours is pretty big because it carries a pretty hefty imager on it, or a camera, and some pretty hefty batteries, so it is quite large. And we use a drone for a number of reasons. One of the benefits of using a drone is that it has higher spatial resolution than satellites. So satellites can have a resolution of tens of meters usually, whereas the drone is able to fly quite close to the ground and get a resolution below a meter. And that sort of is also a benefit that fixed wing aircrafts can have, but usually it's a lot more of an endeavor to do those flights. So there's an added benefit of using a drone, which is that we can collect data whenever we feel like it. For example, the National Ecological Observatory Network, or NEON, has a field station at Blandy, and they collect fixed wing aircraft imagery once a year around July. So there's this really high quality data set from them, but it only provides images for July. So we opted to use a drone so that we could look at plants at the very beginning of the growing season, all the way to mid-growing season, and then into the fall as leaves lose their greenness and drop at different times, hoping that that would aid in detection of plants. And then there's also the added benefit of having continuous ground coverage. So what I mean by that is, normally if a traditional approach to locating invasive plants is to do ground surveys, where you can sort of walk in a line, get an idea for what's right there where you're walking, but not necessarily seeing what's 10 meters in either direction from where you are. So the drone gives us a really continuous coverage of what is there on the ground. Okay, so the other part of this, and I mentioned before, the drone carries a pretty heavy camera or imager, and we call this imager a hyperspectral imager. So I'll kind of walk you through the concept of hyperspectral remote sensing. So remote sensing in general is looking at an object and figuring out information about that object without actually touching it. So any kind of like Earth observation from space would be considered remote sensing. And the drone similarly provides us with an approach to remote sensing. And hyperspectral remote sensing provides us imagery that has, it basically measures reflectance over really narrow bands. So we're able to see details at a less than 10 nanometer spectral resolution, as opposed to satellite imagery, which is often multispectral and just has like a few broad bands. So to get more specific about the imager that we use on our drone, our bands are about 2.5 nanometers wide. So we get a lot of detail spectrally. We get a lot of information over a lot of different bands or wavelengths in the electromagnetic spectrum. So plants generally have what we call a spectral signature that looks like what you see on this slide. So basically, what an imager sees, whether it's a satellite or a drone, is the reflectance of visible and near infrared and sometimes shortwave infrared wavelengths that are coming back off of that plant, that are reflected off of a plant. And all plants have this general shape when we look at the reflectance over multiple wavelengths, where you see kind of a peak in the green areas of visible light and a little dip in the blue and the red areas of visible light, which checks out with what you learn kind of in middle school when you first are learning about photosynthesis, how leaves absorb red and blue light and reflect green light. And we see that in plants' reflectance. And then there's this really big spike that healthy vegetation has from red wavelengths into the near infrared wavelengths. And then plants also generally have this little dip in the higher near infrared wavelengths that's due to their cell structure. And then there is also this pattern here in longer wavelengths in the shortwave infrared. So just in general, that's what plants look like. But plants can also have subtle differences because of chemical and structural traits that can vary, presumably, by species. So this is actually a graph made from data that I collected in 2020, so last year, in April and in June. So this kind of demonstrates these subtle differences that we can see among species. And those subtle differences are caused by differences in chlorophyll content and carotenoids, so just like pigments of plants. It can also tell us something about plant stress, like if there is a drought, and the nutrient content of leaves. So we see these differences that we presumed would be there. And they, to give you again, like a review of what plants generally look like, there's generally this pattern of low reflectance in blue wavelengths and in red wavelengths, a higher reflectance in the green wavelengths. But we see these, there are definitely subtle differences caused by differences in pigments. And then there's this steep increase into the near infrared. And this is, we see kind of the same patterns in June. And these are a few species that grow at Blandy. The thick line that you see is the average for that species, and the thinner lines on either side are kind of our sureness that the mean lies between those lines. So in April, you can see here, this is autumn olive, this light green line, and you really see differences in autumn olive in this blue region, where the blue reflectance is higher than most of the other species. And you see it also in the red region. And even the green, the reflectance is pretty high relative to other plants. Another plant of interest that we're focused on, out of those 3 focal species that I mentioned before, another one is buckthorn, which is colored in black on these graphs. And then the 3rd species of interest is tree of heaven, which is colored in this kind of purplish color. So you can see that there are definitely differences, but also there's quite a bit of overlap between these plants. So it's not necessarily always ideal, but again, the drone allows us to fly at different points in the growing season. And so we have actually pretty good separation of autumn olive in April, for example, but maybe not tree of heaven in June, and maybe not buckthorn in either of these months. There might be a better time in the growing season where we can separate those species out from other plants. So that's one of the questions that we're aiming to answer here is, when can we separate out those plants? Okay, so that's some kind of nitty-gritty technical stuff. This is just zoomed in on that increase from the red wavelengths into the near infrared, so that you can see a little bit more separation if we just zoom in on that steep increase. So like I said before, the overarching question is, how do we detect these invasive plants? And the one that I'm going to focus on today is autumn olive, because we've had some success with that so far. This is a really slow process, like Rod said. So that's about as far as we've gotten so far. But the goal is to also answer this same kind of question for tree of heaven and buckthorn. So I know I've mentioned before that we have a drone. It has quite a heavy imager on it. This is that imager. It's a hyperspectral camera, and it collects images from 400 to 1,000 nanometers. So if we actually go back to this picture, it actually only collects data, reflectance data from 0.4 micrometers to about 1 micrometer. So we don't get the added benefit of seeing all of this reflectance in the longer wavelengths of shortwave infrared. The drone, I said before, also, it has bands that are just a few nanometers in width. So it actually collects data over 270 bands. So basically what that looks like is we're collecting what's essentially like a digital photograph, where you can see the balance of the red and the green and the blue and the light and the dark. But instead of just having the balance of those few colors, we instead have data for 270 bands. So there's a ton of information in every single pixel. And the pixels, because the drone can fly so close to the ground, are about 3 centimeters in size. So 3 centimeters by 3 centimeters, which is essentially the size of a leaf. So what we're getting from this drone imagery is really fine spatial resolution. I know I mentioned before, satellites collect data at tens of meters. This is collecting data at a few centimeters. So it's super fine spatial resolution as well as super fine spectral resolution. So we're getting a lot of detail in every picture that we collect. And the drone flies a planned flight over a field. It flies in a straight line collecting images. It turns around and continues to fly in a straight line collecting images, which gives us a photo that looks like, I have to go all the way back to the beginning, that looks like this. When we combine the rows together that the drone has collected, we can basically get a whole really nice looking photo of a whole field. But it's more than just a digital photo. It has information about the reflectance in 270 bands. Okay, so with these data, I collected data from 2 fields, one that serves as a training data set to produce an algorithm to detect autumn olive. And in the future, it will also produce an algorithm to detect ailanthus and buckthorn, and then a second field to test their accuracy. So the one field, totally separate from the other field, trains the algorithm, and the other one tests its accuracy. So what I'll be talking about today is the outcome of 2 different models from 2 different dates. I showed you the data earlier from April and from June, and those are the 2 dates that I am examining here and talking about the effectiveness of those models. So I produced 2 models from this training field. One that is a super simple model that just actually uses 2 bands. One in the blue region. I pointed out to you before that the autumn olive reflectance data looked really different from other species in the blue region. And then 1 band in the red edge, which is that steep increase from red wavelengths to near infrared wavelengths. So it just uses 2 bands. And the reason I opted to do a super simple model like that is so that for mapping purposes, it would be very simple, relatively speaking, and not very computationally demanding. Because like I said, we have these pixels that are a few centimeters in size. we have 270 pieces of information in every pixel that can really quickly get out of control when you're trying to analyze all those data. So I just produced a simple model using 2 bands so that then when I look at the entire image, instead of loading 270 pieces of information, I'm just loading 2. And then I also did a more complex model that evaluated it for all the bands, which is really nice if you have the computing power to be able to do that, but not that nice or practical when you have to do something like produce a map. And like I said before, I did this for 2 dates, April and June. So after I produced the models on the training field, I tested them out on the test field. And so I looked at 46 individuals. Those individuals are trees and shrubs that I knew their identity from being out in the field. So I, in applying the model, pretended like I didn't know what the identities were to see if the computer could figure out their identities, and then compared that to the identities that I knew from visiting these locations in the field. Out of those 46 individuals, 34 of them had leaves in April, so 12 of them I didn't include in the evaluation. And 11 of those in April were evaluated, and 13 of them were evaluated in June. And after I evaluated those models, any individual that had a probability of classification as autumn olive over 80%, any pixel that had that, I then said, okay, this is probably autumn olive. And if a single tree or shrub had at least half of its pixels being very likely to be autumn olive, I presumed that the computer is recognizing this individual tree or shrub as autumn olive. So these are the outcomes of that simple model that just used 2 bands for each of those months. So like I said, in April, there were 34 total individuals that I looked at. 11 of those were autumn olive, and the computer picked up 4 of them, but missed 7 of them. So out of those 11 individuals, it said, I see these 4, but I missed these 7. So that's not really great. However, there is the goodness that it didn't falsely identify anything that wasn't autumn olive as autumn olive. So the false positives, there were none. That means nothing that wasn't autumn olive was deemed to be autumn olive by the computer. And the true negatives were individuals that are not autumn olive that came up as not autumn olive. So that's a good thing. At least we didn't have false positives. Then in June, when I looked at all 46 individuals, because they all had leaves by then, there were slightly better results for the autumn olive detection, where out of the 13 individuals, 9 of them were detected and only 4 were missed. And there were, again, no false positives. So that's a pretty good outcome for just a simple model that used 2 bands, not very computationally demanding, and how that went. Okay, the more complex model, which used all the bands, had better results, which isn't very surprising. Out of the 34 individuals, there were 9 autumn olive individuals that were detected and only 2 missed this time, as opposed to the 4 and 7, respectively, with the simple model. And the same outcome for false positives and true negatives. So that's good. So there were definitely better results with detecting autumn olive. And then in June, we had even better results where there were all 13 individuals were detected by the computer. And the only misclassification was a single false positive. So that's pretty good. But that's not surprising because it was a model that used all the bands. The good news is that it's possible, because differentiating a bunch of green plants from each other in a photograph is not easy. It is really quite difficult. But using this hyperspectral imagery shows a lot of potential to be able to do this. This is an example. I know I said before, it is much easier to be able to map the location of these things, map the probability that a pixel's autumn olive using a really simple model. So this is using just the 2 bands. And like I said before, this was from 46 individuals. So there was just like a subset of the trees and shrubs in this field that I actually visited. So anything that is circled in pink, I actually haven't visited yet to decide, is this really autumn olive or not? But the things that are circled in blue, I have confirmed that those are autumn olive. So there are shrubs or trees that we're seeing here that I haven't figured out if they are autumn olive yet. There's a good chance that they are because of the outcome from those 34 individuals. So that shows promise. But the more complex model would definitely be much more computationally demanding because it would be about 135 times more data to produce a map. And I don't really have the computing power to actually produce a map of that, unfortunately. So the next steps are to continue to examine the spectra, not only of those other 2 target invasive plants that I talked about, ailanthus and buckthorn, but also to look at different points in the growing season. So last year in 2020, I diligently went out every couple weeks, early in the growing season and late in the growing season. So basically during April, May, and June, and then in October and November, I went out every 2 weeks and collected data for these fields. And so we should be able to look at all of those and figure out when's the best time in the growing season generally to detect these invasive plants, to be able to differentiate them from their surroundings. And then the other thing that I would like to do is try to extend our really fine resolution, our fine spatial resolution data to a coarser resolution data set from the National Ecological Observatory Network, who, as I said earlier, have a field station at Blandy. They collect data in around July of each year at a 1 meter resolution, which is still really fine spatial resolution, especially compared to satellite data. And their data is also hyperspectral like mine is. So it would be really nice if we can scale up spatially from my data that's drone based and about the size of a leaf to something that's maybe more on the branch scale or even the shrub scale. So, and then also cover more space to be able to do this at the landscape scale. That would be really obviously ideal for someone like a land manager with a really big piece of land to be able to look at that and say, oh, here's where I have autumn olive or ailanthus growing. So that's all I have for today. And I guess now we can take some time for questions.
Yes, thank you, Kelsey. Yeah, we can take questions from the audience. Are there any questions out there? Or from any of our panelists? Well, I did have a question about your work, and I'm curious of what got you interested in learning about drone technology and connecting that to invasive plants.
That's a good question. So I first learned about hyperspectral remote sensing from Shi Yang's class. Shi Yang is that second name here on this slide. And he is one of the professors at UVA who is on this project and on my committee for my PhD. But he was kind of the first person to ever teach me, well, one, about remote sensing, right? That you can infer information about something without coming into contact with it, but also that you can get such detail and make predictions about plant species characteristics using these. It's really just like numeric data. And as a math person, I was like, oh, that's so cool, who's also an ecologist. That really resonated with me. So that sort of is where I got into remote sensing. And as for the invasive species detection, the work that I did before focused a little bit on native plants versus non-native plants and invasive plants, and what their impacts have on things like nutrient cycling, productivity of ecosystems. And so I was already kind of interested in what are the ecological impacts of invasive plants, non-native plants. But this project was really the brainchild of Rod and Shi and Howie, Rod from PRISM and Shi Yang and Howie Epstein from UVA. They came up with this project, and now I'm the person running with it.
That's cool. And another question that I had, and then we do have some other questions that have popped up from the audience. So this gets to where, like, yes, you can detect tree of heaven or any other particular species with good accuracy. How would this be packaged and sent out for like land managers to use? Would it be like an app on your phone or software or like integrated into ArcGIS?
Yeah, that's a good question. What I strive to do is, well, I do all of my data analysis in R, which is an open source program. And my goal at the very least is to make this as open of a process as possible. So it might look like software eventually. It might be, you can put in a picture and the software evaluates it for you. It might be sort of just like some code that is already written for you and you just run it, which would ideally not require a lot of understanding of coding or programming, right? But I'm not sure exactly what it would look like. I would envision something along those lines. Maybe not an app on your phone, just because it's so much data.
Yeah.
But maybe an open source program that you can run.
Okay. And kind of a follow-up to that from Andrew, one of our panelists today. He says, this is really impressive and it's coming along nicely. How long do you think it will be until this really comes into its own and becomes something for large landowners to use for invasive species detection or combined with LiDAR canopy height data? So what is the timeline for developing this kind of technology?
I think in another year, I should have like a good understanding of what it takes to be able to differentiate plants from their surroundings, at least at Blandy. And then maybe at that point, it needs to become more of a team effort where someone goes out in a different area, maybe like 100 miles away, and assesses whether or not we can detect those same plants in their surroundings at that point in the growing season. So it might take a little bit of, I really appreciate Blandy in that it provides me a really heterogeneous environment to be able to detect these invasive plants within. But I also know that Blandy has its own group of trees and shrubs and grasses that grow there. And that isn't necessarily what these plants are growing in everywhere else in even the Blue Ridge area. So it might take a little bit of, it'll take another year probably from me to get these algorithms all in line, but then maybe another year or something, depending on how much time people can devote to it, to actually testing it out elsewhere. And what I would really like to see is if we can apply this at the landscape scale, like I said, with something like Neon, or if we can even find really wide swaths of land that have these, and even use satellite data to do this. That sort of is a pipe dream at the moment, but is really one of my long-term goals, is to try to figure out how we can do this on a pretty wide scale, because I know that even though invasive plants are definitely an issue at Blandy, and we want to know where they are exactly and how long they've been there, they're an issue all over. And that's why everybody's here today. So yeah, Andrew, I think that's a really good question, but hopefully that sheds some light on it.
Yeah. So we have a question from Michael, and Michael wants to know, how much computing power would you need for the complex model calculation?
To make the whole map, that's a good question. And I'm not sure. I know what's kind of complicated about producing the map using all of the wavelengths is when I try to do it in R, I just can't. It won't let me look at a map with all the wavelengths. So it's something that I need to figure out. I think I don't know if she is still on this call. He was here for a minute. But I think that there's a way that we can do it. I just need a little bit more guidance and assistance to be able to do it. But I think it's possible. I just haven't done it yet. So I think it's possible.
Mm-hmm. Okay, well, great. So we have several other questions that have popped in. So I'll go to, so can you elaborate a bit on how you process all of your data and generate the algorithms for detecting which plant species you're dealing with?
Can you repeat that? I'm sorry,
Yeah.
I was glancing at the chat.
That's fine. Can you elaborate a bit on how you process all of your data and generate the algorithms for detecting which plant species you're dealing with?
Yeah. So what I have done so far is I have these individuals. So first, I start with my training field, my training data set. And in that training data set currently, what I have used so far is 64 individual trees and shrubs whose identity I know. So from those 64 individuals, I look at the image of them, and from each individual, I pull out the reflectance spectra of 15 pixels from that individual. So basically, I'm creating like a subset of data from the entire picture. So from that, then, I recode the data into, for example, if I'm trying to figure out if I'm trying to detect autumn olive, I recode each data point into either yes autumn olive or no autumn olive. So everything that's autumn olive, I say yes. Everything that's autumn olive, I say no. And those also equate to 1 as a yes and 0 being no. And I equate that to then the probability that a pixel is autumn olive. And in that training data set, there's 100% that those autumn olive pixels are autumn olive, and there's 0% chance that those non-autumn olive individuals are not autumn olive. So that sort of is like where the data begin. And within those, there's 270 data points. And the way that I opted to make the simple model is I performed a partial least squared discriminant analysis to see how those pixels all separate out from each other and what wavelengths seem to be important, and just applied those important wavelengths in the linear model and simplified it to make it like as simple as possible, but also explanatory as possible. So that's kind of how the simple model works. And for the more complex model, I used a partially squared regression where I used all 270 wavelengths as a predictor of the probability that a pixel would be autumn olive, with those probabilities being 1 versus 0. So that's kind of how those two models work. And then once I have the algorithm, once I have like the model produced from the training data, I take the testing data and basically plug it into those models, and then get a value, like an output, that's also for the probability that each of those pixels are autumn olive. And then I can evaluate, did this model effectively detect this individual? And I'm happy to take like a follow-up question from that.
Is there a follow-up question from the audience on that one? Not coming through. So we have another question from Rob, and they ask, can this work in half of the year when there are no leaves on a plant effectively, or is it just during the growing season?
That's a good question. So you can do data collection whenever, and I actually have collected data in March from before any leaves were out, so that it was basically bare trees, any standing dead leaf material, and then any evergreens. And that's it's possible to use to detect, but because leaves cover so much more, leaf canopies cover so much more space than just the branches, I think that there's more likelihood of being able to detect these plants, unless there's something really special about stalks that you can see. I know one plant that Rod has talked about before was wineberry, with its sort of reddish stalks, and how those are visible in the winter when there are no leaves on it. Or Japanese honeysuckle that has its leaves still in March when other things don't have their leaves. Those are plants that can benefit from doing data collection in the other half of the year when there are no leaves. But otherwise, I would say leaves are definitely the key to differentiating these species, probably more so than the bare branches.
Okay. And I left off part of the question, and they specifically meant, can you detect like rose fruits, branching styles, monocultural stand appearance? I think you started to get at some of that in your answer, but do any of those things change your answer a little bit?
The branching style might actually be an interesting feature to examine with LiDAR, which is one of the things that Andrew, I think, mentioned in his question. So maybe, yeah, if you're taking into consideration, I'm mostly focused on what are the differences in the leaves spectra that allow us to differentiate these. But there's a possibility that you could use LiDAR and, canopy geometry and branching geometry, like you said. So yeah, it could be possible for sure. There's a lot of different approaches to doing these things.
Okay. And then someone just asked, would fall colors help with the identification?
Potentially, yeah. So especially if you were just using like a regular photography, and you could kind of look at different points in the fall and get an idea for what changes colors when, how the colors look, it would especially be helpful there. In our case, we get even more data than just the visible color, which, yes, absolutely, that can help to detect an invasive plant. But we're also getting the benefit of so much information being in these images that we can even make inferences about, the chemical composition of a leaf at a given point in the growing season. So I think, yes, fall colors help, and probably also changes in chemistry that take place at the end of the growing season across different plants and how those differ, how plants kind of lose their greenness at different points can certainly help, which is why I collected data every 2 weeks in the spring and in the fall, and once a month during the summer, because there isn't as much change taking place during the summer months, you know, from June to August. But then things kind of pick up again when we have pretty quick, swift changes in leaf chemistry and the way our leaves look at those points in the growing season.
Okay, and following up about elaborating on how you process all of your data and generate the algorithms. Garrico, I'm sorry if I'm mispronouncing your name, said that your explanation helped a lot, and then followed up with, have you considered loading your data into a database system such as HADOP, H-A-D-O-O-P, instead of loading into R, which will quickly run into memory limitations?
I have not, but I will.
Yeah,
Spell that for me again, Beth.
It's Hadoop, H-A-D-O-O-P.
Okay, I'll look into that.
I'm not a technology person. Yeah, thank you for that that really excellent exchange about the processing the data. And then we have one more question and comment, and they say, this is quite a sophisticated drone setup and imager. How hard is it to fly, and are there any qualifications to fly it?
So yes, there are qualifications to fly it. I had to get my drone license, which I had no background in any piloting type things or any air travel piloting before this. So I did have to study for that test. And it's really just if you're flying a drone for your job rather than for enjoyment that you have to get your drone pilot license. However, the drone basically flies itself. Once we're out in the field, I create these planned flights for the drone to do, where it flies in a straight line at a set speed. And the reason that it does this, that it has to fly at a straight line, at a set speed and at a set altitude, is for the sake of the imager. Because the imager is pre-programmed at the beginning of the flight, and I tell it how fast, how high the drone is flying, and that tells it how quickly it should be taking pictures. Because the way that the imager works is it's taking pictures, you know, every millisecond along the flight, so that it's only recording a picture that's like a row of pixels, and then it pieces all those pixels together. So it's kind of a complicated process to get the imager and the drone flight planning stuff to all work together. And that took a bit of trial and error. And by a bit, I mean like 6 months, just to get those things to align so that I got pretty pictures like this one on this screen. And so once you have the hang of that, it's not too bad. The most complicated thing isn't flying the drone, but getting the imager and the drone to work together and take pictures during the flight plan at the right frequency.
And then I have one last question that's maybe a little quirky. Does your drone have a name, and does it have a personality that you projected onto it?
Yes. So the drone, well, I uncreatively named it Droney, but people have really embraced Droney. Even my advisors refer to it as Droney. And I have a love-hate relationship with it, I would say. Like some days it's very cooperative and it does what I think it should do. And sometimes it doesn't. Rod I don't know if Rod remembers this. It probably wasn't as upsetting to him as it was for me, but I spent a whole day doing flights at Blandy, and it just wouldn't record data ever. So when Droney does things like that, I don't appreciate it. But for the most part, we've learned to get along, and I would say we have a good relationship now. It's pretty cooperative.
Well done.
And we did have another question come in, and this will probably be your last question. So Laura asked, how much longer will you be conducting this research at Blandy? And then any potential opportunity to observe a flight?
Good question. I hope that I'm done collecting data, but because I collected data so frequently last summer, but there's a good chance that I will need to collect more data, whether it's for this project or just for some one-off thing that maybe somebody wants hyperspectral imagery over a field. Since I'm the only person at Blandy doing that they talk to, there's a chance that I would be there. So, and you could absolutely observe a flight if that's up your alley. So yeah, there's a good chance of it, but nothing on the books yet.
All right. Well, Kelsey, this has been fantastic. Your research is fascinating and is very exciting from a land manager perspective. And when I worked in Indiana, we started tinkering with using drones to detect like royal polony and things that were really showy in the forest canopy. And we went back and did some ground truthing, and it was a really great tool, especially in really rough terrain. We knew where we were going, and it was really neat. So it's fascinating and neat and exciting to see that you take that technology and take it to the next level. And we're thrilled that you were able to join us today and answer all of our questions.
Yeah, thank you so much.
Thank you.
Yeah, well done. Thank you.
Yes, agreed. Props from William and Andrew. Okay, so our next presenter is Andrew Rohrbaugh. And Andrew, if you want to go ahead and cue up your presentation, and I'll make the introduction for you. Andrew grew up in South Central Pennsylvania and received his bachelor's in environmental studies at Gettysburg College and a master's in biology at Shippensburg University. He now works as an ecologist for the Pennsylvania Bureau of Forestry, where he helps protect native wild plants and manages the invasive plants that threaten them. In his spare time, he enjoys gardening, hiking, hunting, and raising his 2 sons to enjoy the great outdoors. So Andrew, welcome and thank you. And we'll learn some about biocontrol. So,
yeah, hopefully everyone can see my screen there.
yes,
okay.
We got you.
Okay. And everyone can hear me okay?
Yes.
Oh, cool. Just want to make sure. Sometimes my computer doesn't always play nice with the microphone. So as you said, my name is Andrew Rohrbaugh. I work out of Pennsylvania's our Department of Conservation and Natural Resources in our Bureau of Forestry. And I'm here to talk about biological control for invasive plants. So just like this picture here, when you look a little more carefully, you start to see that there is a spotted knapweed root weevil there. And I always like to start off pretty much all of my presentations with a little bit of background. In Pennsylvania, our Bureau of Forestry is the jurisdictional agency for native wild plants all across the state. And we also have 2.2 million acres of state forest land. So my job is basically to help manage rare, common, and invasive plant species on our state forest land, and also help protect native rare plants across the state. And unfortunately, my jurisdiction doesn't technically include invasive plants for that, but obviously they're one of the main threats. So I work a lot with some of our other agencies and organizations to try to fill that gap. And I will admit going into this that I am not an entomologist. So a lot of our biocontrol species are insects, and I'm not an expert on those, but it's something I've come to learn a lot more about as I look at these from a plant conservation perspective. So we have in Pennsylvania, and I'm sure Virginia's numbers are somewhat similar, we have over 3,000 plant species. About 2/3 of those are native. And about a quarter of those in Pennsylvania are what we term our species of special concern. So those are species that we have some reason to think that they're uncommon in Pennsylvania. That might just be because there's not a lot of habitat or range in our state. But obviously a lot of our plant species are under threat from things like past and present land use, altered disturbance regimes and lack of fire, deer browse in particular, and of course, invasive plant species. So those are the reasons I'm in this area of interest for me for biocontrols, because I want to keep things like that yellow-fringed orchid on the landscape as part of our ecosystems in Pennsylvania and across the greater Mid-Atlantic. And of course, probably most folks on here don't need this, but I leave it in anyways, just in case there's anyone who doesn't know. Invasive plant species that's not native, has not evolved in this ecosystem, this area spreads quickly and aggressively and causes some kind of harm, generally economic or environmental. So we're looking at things like the Phragmites stand on the bottom left or the garlic mustard on the bottom right. And of course, like I said, the reason why we're not appreciative of these invasive plants is because they're displacing our native plant species, and that has harmful effects on our entire ecosystem and all the ecosystem services that come from the native plants and animals. So I love this picture here, and it basically just shows a really great overview of a typical invasion curve. I like to tell people you can look at this in terms of the acres infested there, and you can think this is the entire East Coast. This could apply to just the state of Virginia. This could apply to a local forest tract, or it could just be your 1 acre backyard. What you basically wind up seeing is a, let's see if I can get the laser point on here. Hopefully that should show up. You wind up with a species getting introduced. There's not a lot of acreage. It's really easy on the control cost to treat it. By the time it's detected, it's already starting to ramp up. And by the time the public realizes it's a problem and starts to get engaged, you're already at a lot of acreage infested, and control costs are being much higher. And of course, not only the control costs higher, but you get to the point where you're probably not going to manage eradication. So unfortunately, a lot of our invasive species that people are aware of are at that stage. They're right there, or if not even higher. So what do we do with those? Well, a lot of our management paradigm for invasive species deals with mechanical or chemical control. You go out and you cut or you hand pull these things. Which can be back-breaking labor, or you use chemical control, so using herbicides. That's, even though that might be more efficient from a labor perspective, more cost-effective, it's still, it's expensive. I posted the picture down here. This shows one district's supply of herbicide for a year for just like one tract where they were doing a lot of invasive management. And of course, you've got to get spray rigs to be able to efficiently use the herbicide. So these are really useful earlier in that invasion curve, depending on the kind of the tract, that acreage that I was talking about, whether you're looking at your backyard or a forest district or tract or statewide. But eventually, these aren't going to cut it with a lot of invasive plant species across an entire region once they're widespread and established. So that's when we start turning to look at things like biological controls. In Bureau of Forestry in Pennsylvania, we have them and we're using them for invasive insects and plants. The nice thing about them is you release them and you walk away. You don't have to constantly be revisiting the site and reinvesting in treatment. You treat one thing and another invasive species comes back. So you're on that treadmill. So it's a relatively cheap and effective way to deal with common invasive plant species. And biocontrols are not designed, a common question, to eradicate the plant species that they're controlling. They're only designed to decrease its growth to help bring it into a level with our ecosystem of other plants. Unfortunately, there are limited options for effective and safe biological controls. There were a lot of what I'd term half bad ones that were released back in the 60s and the 70s and far beyond that even in time. Plant species that were brought in for biological control without really thinking through how effective they would be or studying potential consequences. And that gave the idea of biological controls a pretty good black eye for a while. And I want to talk about that in a second, but just for examples here in Pennsylvania, I'm very sure down in Virginia as well, we're looking at biocontrols for ailanthus. We've got ones for hemlock delgid, gypsy moth, purple loosestrife, mile minute, and spotted knapweed. So looking at some of those, that black eye that I mentioned, the cane toad is a great example. This was introduced in the 1930s in Australia. There was pretty much almost no research into it. They just knew that cane toads had helped with some beetle impacts on sugarcane in Hawaii. And it was just a real fly by the seat of your pants operation before planting. The wrong beetle species to be attacked, the wrong type of habitat. And of course, toads are not a specific predator. So of course it caused other problems. But things like that really gave biocontrols a bad name. Nowadays, we have a much more rigorous process for biocontrols. Where you're required to get approval from the USDA's APHIS, Animal and Plant Health Inspection Service. There's a technical advisory group that helps run those requests for introducing new biocontrols. And I will fully admit to not being an expert on any of this, but from my research, you have to enter a lot of data into these research efforts. You need to look at not just the range of the biocontrol, the potential range of the biocontrol you're bringing in. You also have to look at the potential range of the host, the plant species you're looking to control. So, and then you also have input from like U.S. Fish and Wildlife Service for potentially closely related threatened species that might be targeted by the biocontrol. And so I like to give an example of that, like Japanese knotweed. I don't think it's spread all the way down to Florida. I could be wrong on that. But when APHIS is looking into a biocontrol species, an insect that might be brought in, they're going to say, well, knotweed technically could spread its range all the way to Florida. And this insect biocontrol could spread its range all the way to Florida. And in Florida, there's some closely related knotweed species that are threatened or endangered. And so now we have to do some host testing on those plant species, even though there's no immediate overlap, but there could be someday. So like I said, it's a much more rigorous, thoughtful process. They also do what's called centrifugal phylogenetic testing. Not an expert on that, but what I can tell you is basically they're looking at that insect biocontrol or whatever it is. They're testing it. Does it eat like Canada thistle? Okay, it does. They're interested in bringing it in. Well, then they start testing it to see if it will eat or reproduce its life cycle on other related native thistle species. And then they start looking from not just species in the same genus, but they start looking at some species from the nearby family and other genuses. And then they do less and less testing as you get further and further away in terms of relationship, until you get something like an oak species. They might test oak just to make sure that it's an economically important species, just to make sure that that biocontrol insect doesn't feed on oak leaves, just as a freak chance. So another way of looking at this basically is, they're testing. Obviously, they want it to feed on the species that they're targeting. Then they test a lot of the species that are in that genus, test a little bit fewer that are in their family, and then they test some of the plant species that are in families of economic importance or in similar habitat. So they do less testing as they get further away in terms of relatedness to the target species. And I will say that, people question biocontrol because those early black eyes. This is a much more rigorous process. It's certainly, mistakes could happen. Things can change in nature. I don't want to say that this is ironclad, but I think it's a much more rigorous process and very defensible. Because, like I said, that's a question people often have. So another question that I often get is, where can I get biocontrols? Folks see a presentation like this and they want to know, well, where can I get that? I have this species. The first answer that I almost always give to people, because oftentimes I get it from mile-a-minute weevils, are make sure that you don't already have that biocontrol species on your property, because these things do spread on their own, and it takes years for them to ramp up and start to show control over the invasive plant. So that's the first thing to do, is go out and really be certain that you don't have it yet on your property. And adding additional releases once they're already established in your area isn't really necessarily going to speed up your control based on the cost of purchasing new ones. But you can, if you don't have them on your property, talk to some of your local parks or landowners that have the biocontrol. If you can spot it on infestations they've got, and see if you can collect some, so long as it's within your state, so long as you're not moving these things from state to state. The New Jersey Philippa Lampi Laboratory sells some of these biocontrols. It's a little pricey, but if you're really looking to get started, that's a good place to contact. And for some of these, particularly the spotted knapweed biocontrols, you can look to some the Western states have some businesses that actually sell them. And I've looked into that, and we've purchased some for our Bureau of Forestry. At least some of them are on the up and up in terms of having all of their federal paperwork to sell these across state lines, all their permits. So that's something to look into. So the first species that I kind of wanted to give people a little information on are spotted knapweed. This is one, luckily, we don't have a lot of out here in the East, or at least we have a fair amount, but it's not as big of a problem as it is out in the Western United States, where they have a lot more dry, rocky pasture land, kind of habitat favored by this species. There are a number of different biocontrols out there that have been released for knapweeds and thistles. I'm not going to get into it because there's a lot of them. Some of them have been released purposefully, some accidentally introduced, some of them have been successful, some of them not so much. But I'm focusing on a couple of the ones that have proven successful out in the west part of the country, but for some reason, maybe it's because economically knapweed isn't as big of an issue out here, didn't seem to ever be introduced here out east, at least not extensively. So the first one that I'd like to highlight is the root weevil. At certain points in this presentation, I will attempt to pronounce the Latin names. You'll have to bear with me if you're a Latin scholar. I probably am butchering different pronunciations, but Siphocoleonis acadis is the root weevil, fairly decent sized little weevil. And what this one does is actually lays its eggs on very large knapweed plant individuals. The larvae burrow into the roots and can destroy larger plants, at least severely weaken them, if not completely kill them. This is one that does not move well on its own. So if you do not have this released near you, this would be a good species to try to purchase. And I have released this one, as well as the next species I'm going to talk to on one of our forest tracks in central Pennsylvania. And I have recaptured this one over the 5 years since I released them. So it has established here in Pennsylvania. The other one that I released in that area was the seedhead weevil, Larinus obtusus. And this one actually lays its eggs on the flower bud, and the larvae burrow into the flower bud and destroy the seeds. So it decreases the amount of seed setting from the spotted knapweed. So you can see how those two kind of work in tandem, one destroying larger plants that are probably the most vigorous competitors and seeders, producers of seed, and one that attacks seeds and decreases the amount of seeds set by the species. I have not recaptured this one on our state forest track that I released it, but I think I've been going out at the wrong time of year to really get good recapture on that. And once again, not an entomologist, so that's a mistake that I made, I think, for a couple years. So just as an example, this is an old reclaimed strip mine, hundreds and hundreds of acres that our Bureau of Forestry acquired. We wanted to protect this site and keep it as early successional habitat, early successional grassland, because it turns out there were a lot of rare bird species using it, upland sandpipers, Henslow sparrows, I believe, short-eared owls, and harriers. So we've been trying to encourage native plant revegetation here, early successional species, and you can see spotted knapweed was quite abundant in some areas. Way too abundant to consider any kind of mechanical or chemical approach. So what we did was we released these two knapweed species, and as you can see, these pictures are a little misleading because even though they're the same location, several years apart, 4 or 5 years apart in this image, you can see that there are some differences in knapweed. Partially, it is actually because of the phenology. You know, if you take this picture right when it's blooming and take this picture just a week or two too early or too late, it doesn't pop as well. But I was doing vegetation plots in these areas, and we did see a decrease in spotted knapweed over those 5 years or so. And part of that might be because this site is becoming a little bit more organic and rich as it revegetates. So is it because of the biocontrols? Maybe not entirely, certainly. 5 years isn't that long for these species to ramp up their populations, but I'd like to think that it's helping to play a role. And we've since moved on to trying to release this on some of our other mineland areas, right-of-ways, places where spotted knapweed is aggressively taking over areas. Another plant species that we've been dealing with biocontrol is mile-a-minute. And so we've been releasing the mile-a-minute weevil. I actually looked at a map to see how far mile-a-minute has spread into Virginia, and it looks like about half the state on EdMaps does not have mile-a-minute yet. You should be very thankful if you're in that half of the state. This one is a real pain. It's an annual vine. It grows very quickly, just drapes over all the native vegetation and smothers it, and it has these nasty recurve spines that make it a real pain to try and pull out. And of course, as an annual, anytime you treat it, you go back out the next year, and you know, if the seed bank's still there, it looks like you've almost done nothing. So it takes a lot of work to keep after this one. Luckily, these little guys in this picture, they're orange. When they're younger, they're black, and they grow this waxy orange exudate out of their exoskeleton. They do a pretty good job of helping us. They were first released in Delaware, New Jersey back in 2004. And the adults kind of hide out at the leaf tips, the growing leaf tips, as you can see here. And this one you can see is just starting to get that waxy covering. They lay their eggs, the larvae burrow into the nodes of the plant and cause severe damage there. I believe studies have shown this doesn't actually decrease necessarily the biomass of the plant, but what it does is, because of that physical damage, makes it grow shorter and bushier, which allows native plants to do a better job of competing with it. So this is one we've been spreading across Pennsylvania. Wonderful little species. We've gotten really good establishment. It moves well on its own, up I think like 10 kilometers a year or something like that, one study showed. So it's doing a good job of what we wanted. We tossed it on the landscape and it does its thing. We don't have to worry about it. It doesn't really like shady conditions, which is fine because mile-a-minute doesn't either. But we have seen, and research has shown, that when you have a cooler or moister summer, That doesn't slow down the mile minutes growth, but it does slow down the reproductive cycle of the weevil. So you wind up having a lot more mile minute growing that year. I think it's also because a lot of the precipitation encourages those annual plants with weaker root systems, perhaps. And so you wind up having booms of mile minute in those years, we found. Kind of seen the same thing with stiltgrass without the biocontrol aspect to it. And this, like I said, it does work well with native vegetation to help outcompete the mile minute in the long run. And I will say, if you are looking to get this established, this is one that I often tell people, you know, it spreads so well on its own, you might already have it. Take a look for it, go out on a cool morning, late in the summer, like August, look at the mile minute you've got, look down at those growing tips very carefully. If you walk up slowly so you don't startle the weevils, that sounds like I'm joking, I'm really not. You walk up slowly so you're not shaking the plants. It's cool in the morning so the weevils are a little slow, and you'll see them out on the growing tips. If you look carefully, you'll also see some of that damage at the nodes, which is very characteristic of the weevils. They will chew holes in the leaves as adults, but so will Japanese beetles and other species. And if you want to capture them to respread them, go out one of those mornings in August, cool morning, carefully, you can pick them off the leaves and just shake them into a coffee can, or you can shake the whole bush of mile minute, the whole vine onto a large sheet of paper and fold it in half and just dump the weevils into a coffee can. Make sure you put some kind of vegetation, either crumpled up mile minute or something down at the bottom. That will help keep some moisture for the mile minute weevil as you take them someplace else and help them survive. But also I found their native defense to predation is just to drop off and hide in the undergrowth when they're disturbed. So if you put something in that coffee can, then they feel, I think, a little bit of protection being under that inside the coffee can, and they're not going to try and crawl out right away, or at least not as quickly. I've moved quite a bit this way across Pennsylvania. These are all the points that we've had known releases, and you'll see that there's some blank areas here. This is kind of approaching the edge of the range of the mile minute right now. I'm sure there's a lot in here, and we just haven't targeted them yet with individual releases. And frankly, I'd be surprised if the weevils haven't filled in most of these gaps here on their own. Another one I'm not going to spend a lot of time on here because this is a little bit older, but the purple loosestrife biocontrols. I looked into, actually, let me back up here. I looked into this a little bit because we have purple loosestrife still in Pennsylvania, obviously. And I wasn't sure when I started this job, you know, how are these biocontrols? Have they worked? So I tried to do a little bit of research, and it seems like Cornell's Stacy Endriss, ENDRISS, is a good resource. She's been working with Bern Blassie to look into their establishment in New York State. And they found all 4 of these species have been widespread and are doing a good job of controlling purple loosestrife. They've shown significant decreases in density of plants, the number of inflorescences, the number of flowers per inflorescence, and an increase in other native plant species in wetlands impacted by purple loosestrife. Yeah, even though these are older biocontrols, it's good to see that they've established, and implies that maybe we don't need to do additional releases, but that's something that you can work out on your own. Looking into those, we have the Galaricella beetles, which are 2 beetle species that feed on the leaves. And these are ones that are relatively easy to see on the purple loosestrife. You'll see them out during the day. The feeding damage is pretty noticeable as holes in the leaves. Here in Pennsylvania, these are the places we know that they were released in the blue. Like I said, I'm pretty sure these are very widespread in Pennsylvania. New York's research leads me to conclude that we probably have similar population distributions of these in Pennsylvania, just no one has done follow-up to check. The root weevil for purple loosestrife, very similar to the spotted knapweed, where it burrows into the root with its larva and kills very large plants. This one doesn't fly particularly well on its own, but it can so it can spread on its own, and it's nocturnal. So I've never seen this one personally. We've had a few releases of that in the blue in Pennsylvania that I've been able to find looking back in our records and other agencies' records. And then the seedhead weevil, the Nanophis marmoratus, which much smaller, very similar to the spotted knapweed seedhead weevil. It destroys the seeds before they can spread. And that one apparently flies very well on its own. I only am aware of 2 releases in Pennsylvania, but I'm sure there have been more. On to Tree of Heaven, obviously with spotted lanternfly and just the general bad nature of Ailanthus. This one's on a lot of people's minds. Here in some of our forestry lands, it's hard to see in this picture, but this is one of our districts where the district conducted a cut, and they had treated all their Ailanthus previously, several years before, and it's hard to see, but a lot of the regeneration they got over tens and tens of acres was just a solid mat there of Ailanthus seedlings. So, you know, here in Pennsylvania, when we look at stuff like this, when we look at the spread of spotted lanternfly, we're pretty desperate for something to help us with this on the landscape. And luckily, of course, as most of you are probably aware, they did find a verticillium wilt that impacts Tree of Heaven. You see the normal looking tree on the left, and the striations and the darker color on the right, which indicates the verticillium infection. Rod gave a pretty good update on some of this. So it was a little more recent than anything I've got, but a little bit of background for those of you that aren't aware. It was originally found on one of our Bureau of Forestry tracks here in Pennsylvania, killing a number of different tree species. You can see some images there. And it's now known across many locations in Pennsylvania. They found it also in Virginia and Ohio. Testing that's been done previously shows very minimal impacts on other trees. I think they showed some impacts on striped maple, Norway maple, and some of the Aurelia species. But oftentimes those were not deadly unless those species were directly injected with the wilt. The wilt is actually great in a large stand of Ailanthus because once it gets into one tree, it spreads through the root grass and often kills almost all of the trees in the stand. The wilt itself puts out spores that stay in the soil. So in theory, at some point, the growing root tips of the Ailanthus trees might counter that fungus at some point. It might also be spread by bark beetles and whatnot, ambrosia beetles that move amongst the Ailanthus trees. But so yeah, it's a very deadly pathogen for Ailanthus. It doesn't seem like there's been any move towards any resistance in Ailanthus to it yet. But the kicker, as Rod already mentioned, is that this one is not approved for general use. The reason for that is anytime you have a microscopic agent, like a fungus, a bacteria, a chemical, that you are using to kill something, technically registered, it technically needs to be registered as a pesticide under the EPA's law. So Rod already mentioned that study is ongoing, and hopefully it'll be done sooner than later, is all I can say there. There is a group out of Europe, CABI, that is also studying a mite that they found can destroy and to form new leaves on the Ailanthus. So they're doing ongoing research with that. But that's pretty early in the research process, I believe. And here are some of the places that we know it exists in Pennsylvania. Some of these are just naturally occurring, and some of these are places where we've worked with researchers to test it on new stands. So part of the problem here is in addition to getting that EPA registration, it can be difficult to get this established unless you really know what you're doing. I know of people that have tried to spread this a multitude of different ways illicitly, and they've had mixed results. So like I said, hopefully that registration process will allow this to be used widespread, and also in a lot more easy fashion and more successful fashion. So some of the species that people might not be as aware of, there is testing being done for some knotweed species biocontrols. There's a psyllid known as Aphelara itadori. There are actually two different varieties of it. One targets Japanese knotweed preferentially, one targets giant knotweed preferentially. So they tried to release this, which was originally from Japan. They tried to release it in the United Kingdom and Canada. Unfortunately, it appears that they didn't have much success, partially because United Kingdom and Canada are actually at a further north latitude than Japan. And so the difference, the shorter day lengths actually triggered the insect that go into diapause, which is basically its body said, oh, days are getting shorter, better shut down for winter, which is not what you want them to do when you want them to be eating knotweed. So they are now currently testing it here in the Northeast. I think they're doing some more work in Oregon as well. And it's a very large project with a lot of researchers involved in it. It was released in 2015 in Canada, like I said, where they did find it overwintered to reproduce, but just didn't ever see a big boom from the population. So ongoing research on this one, from what I was able to find out from my research, they actually went back to Japan to collect a new population of the psyllid in 2019 because they'd been working with the ones that they'd raised in the laboratory and bred in the laboratory and bred in the laboratory for years and years. And they were concerned that they basically inbred these insects. There's a population bottleneck in terms of the genes. And so they're hoping that by collecting new populations from Japan, they'll be getting more genetic diversity and also helpfully select for psyllids that will go after younger knotweed plants and really damage them before they can get big and do a lot of damage. So obviously, we want to get them before they get to this stage in their development. I don't have a lot more information on the exact latest research or where they're at, other than what I've told you so far. But if you were looking for more information, Fritzi Grostad at Oregon State University would be a great contact. Garlic mustard is another species they've done research for biocontrols for. This is a root weevil, so similar to the other ones, lays its egg. The larva burrows into a garlic mustard root and destroys or severely weakens it. This is an interesting one. You're going to hear this repeated several times, but it's still under review in the United States to make sure we don't have any negative impacts from it. But our neighbors to the north looked at it and thought, we have no concerns in Canada. So they went ahead and released it in 2018. There's no judgment there. I think it's interesting. So sooner or later, assuming that this species acts well as a biocontrol, we should be seeing it here in the US. Actual releases here in the US, we'll just have to wait until we've done our proper due diligence here in our country. And like I said, that's no judgment towards Canada. They did their full review and everything like that. Didn't seem to have any issues. Here in our country, we had a few concerns still that held this up. Like I said, I'm not an expert on any of that. One issue is that it appears that the populations they were rearing to release actually had an infection of some kind of parasitoid. So you can understand that if you're releasing this biocontrol to act as a parasite on garlic mustard, you don't want your biocontrol to have a parasite on it that slows its growth in its population. So they're actually taking the time to try to rear generations as adults and then release their eggs to avoid spreading the parasite. I'm not 100% up on all that, but that seems to be one issue with this species. They do think it has established in Canada, but they're not 100% certain yet because it's so recent. And obviously, for anyone that's not aware, thankfully, maybe in your case, not aware of garlic mustard, you know, you look at the basil rosettes. This is a biennial plant. You have those basil rosettes on the right, and then the flowering plant the second year on the left. And you wind up getting things that look like this. There has been research that shows that perhaps garlic mustard isn't the villain that we thought it was in terms of eradicating spring ephemeral wildflowers habitat. Bernd Blossie's done some research with others that show that it's more of an impact from deer and worms than it is the impacts from the garlic mustard itself. But you know, I think probably not having the garlic mustard is probably a good thing in the long run. So I'm rooting for that root weevil. That's terrible. I didn't mean for that to be a pun. I apologize for that. So anyway, so that's the status of that species. Another one that is being studied is stem-boring moth, the Arcanera moths for Phragmites. This was also one that, like I said, was released in Canada in 2019. One of the reasons it's stalled here in the US is because of we have the native Phragmites australis subspecies, americanus, which is very rare, particularly here in Pennsylvania. I don't even know if Virginia has populations of it or not. But so the concern was that these Arcanera moths might impact those very rare American subspecies of Phragmites. They have done some subsequent research, and they found that while the Arcanera moths will lay eggs on our native subspecies, they don't prefer to. And also the eggs don't survive and hatch, and the larvae don't survive as well on our native subspecies. So that's a point in favor of releasing these in the US. Another thought in terms of releasing these is the subspecies, the European subspecies australis, is really, really, really aggressive and is pushing out our native subspecies a lot of different places. So it might be a moot point as to whether we, you know, let the non-native Phragmites, the invasive Phragmites, spread and wipe out our American subspecies, or whether these moths do a little bit of damage but help to protect other habitat for other species. Like I said, released in Canada, so we'll see how this goes one way or the other. That was a relatively recent release, so we won't know for sure how things are going there. So once again, for anyone that's blessedly unaware of Phragmites infestation, that's what it looks like. We're also doing research on swallowworts, pale and black swallowwort, as you see pictured here. Here's what an infestation looks like. And very close, or closely related, same family as our native milkweed. And in fact, there's been some research that shows it can act as a population sink for monarch butterflies, which might be confused and lay their eggs on it, but the monarch larva cannot survive on the swallowwort leaves and die. So the biocontrol for this one they're looking into is another moth species, Hypena opulenta. Once again, approved for release in Canada and the US in 2018. And the larvae eat the leaves. The problem with this one is that even though it does appear to be doing some damage in shaded conditions where it's released, it is overwintered but not established. And the real problem is that all of them appear to have a pathogen, which came in from its native range, kind of the same thing as garlic mustard's root weevil. You know, we brought in Hypena to try and eat the swallowwort, but this pathogen is damaging the Hypena populations. In this situation, we don't have the option to go back and try to collect more Hypena opulenta moths because the main collection site is in Ukraine. So that's a no-go right now, unfortunately, on a variety of levels. But if you want more information on this, Lisa Tewksbury and University of Rhode Island would be a great resource to try and reach out to and get in contact with. And there is research that's being conducted right now on Gallirusella beetle to attack European water chestnut. And seems like one of the sticking points on that one right now is that the Gallirusella will feed also on our native water shield, Brasinia shriberi species. That might seem to indicate that it should not be released here in the US. However, the Brasinia actually grows with that Gallirusella beetle in its native range and seems very tolerant of a lot of insect damage, not just the Gallirusella feeding. So it might be the kind of thing where, you know, it's not actually that much of a negative impact on our native water shield to bring that Gallirusella beetle over to deal with the impacts and competition from European water chestnut. So that's something that I think they're still looking into and still trying to come up with a decision on. I did want to note too that recently down, I believe in around DC, someplace maybe in Virginia, they have observed a new European water chestnut species, Trappa bispinosa, and I have no idea whether the Gallirusella feeds on that one as well or not. So here's some additional resources. You can actually, I know this is a bit of a long, lengthy HTML, but you might be able to find a newer version of this. It's basically the Mid-Atlantic Invasive Plant Council's Biocontrol update that is available online. So you might be able to find that a better website at this point via Google. I forgot to update that before this talk. There's the Biological Control of Invasive Plants in the Eastern United States, which is a really great publication or a full book on different biocontrols. Particularly useful if you're interested in looking at some of those thistle and knapweed biocontrols that have been either accidentally or purposely introduced and what their impacts may or may not be. Cornell's got a great website on biological controls. And of course, I really highly recommend looking at that technical advisory group for USDA's information on this. And so I will come back to this slide in one moment so that people can keep taking notes here if they'd like. But I would also like to just throw out there, not because I need my picture here with my family, but I want to make sure that my email address is there if people do have questions. Like I said, I fully admit to not being an entomologist, but this is something that I find very interesting and very important for my job of managing invasive plants across Pennsylvania. And so I think that hopefully I've done a good job of giving some general background for everyone here, as well as once folks have gotten my email here, as well as some resources for folks to learn more. So with that, I will stop droning on here. And yeah, that's about it for me. Thank you, everyone.
Well, thank you very much, Andrew. That was a really nice overview of what could be happening in Virginia, and that there might be some biocontrol options out there for the average homeowner. So, so there were a couple questions that did come through during your talk, but then you answered them during the course of your presentation, particularly about the garlic mustard biocontrol. So if anyone else has a question they would like to ask Andrew, pardon my cat, please send it in the Q&A, or if you'd like to raise your hand. And no questions.
I'm going to end sharing here now.
Okay. Well, we did get a comment from Rob, and he said it was informative and experienced presentation. Thanks. So nice job, Andrew. All
Andrew,
right.
are there others that you're aware of that are being worked on, but they're kind of lurking out there? I mean, any updates on things like stiltgrass or any of those things?
Yes, and I have a couple slides that I didn't get into that I held in reserve because I'm even less knowledgeable about those. There are folks that are looking into Russian olive. There's a galling mite that attacks the flowers and shoots of the plant. So there is research being done on that. Folks are looking into oxeye daisy. There is a root feeding moth and also a fly that attacks that one. It's a little harder to find information on some of these, and they're a little bit more in the early stages, which is why I didn't get into all these, but I'll run through them real quick here. There is a shoot boring weevil that they're researching for common tansy. And when I say they, it's different researchers that I found. A lot of research goes on with the CABI group over in Europe, CABI. Kogan grass and Chinese privet are also, big topics in the Southeast for folks researching biocontrols. All I know is people are researching that. I'm not aware of them finding any specific targeted species yet. And kind of similarly, folks are looking at small carpet grass, Arthraxon hispidus, lesser celandine, and Japanese stiltgrass are all being targeted for research. Japanese stiltgrass, they did find bipolaris fungus that attacks it, but I think it was one of those things where it was just not specific enough that anyone thought it could be targeted directly to Japanese stiltgrass and not hit, commercial, agriculturally important grass species. So I don't know that research went anywhere. I could be wrong on that. If anyone has different information, please feel free to speak up. They did just find a rust fungus in Asia that I know they're looking into, but once again, still very early in the stages. I hope and pray for a Japanese stiltgrass biocontrol every day, at least during the growing season at my house and out in the woods here in Pennsylvania. So.
Yeah, no kidding.
So once again, if anyone has any information, that's all very, rudimentary knowledge. So.
Yeah. So nothing for bittersweet. No. Sorry, Norma and everybody. That's a disappointing.
Yeah, I have one other curiosity question, which is, all this work seems to be focused on bringing insects in from the native ranges of the plants and finding ones that are very host specific. Yeah, has anybody ever tried taking the insects that infect closely related native plants and putting them in a place where they either have to adapt to eating this foreign plant or die?
I don't think I've ever heard of anyone doing that. It's an interesting idea. I just wonder whether you'd get a microevolution taking place that rapidly.
Well, right.
And I do. I mean, it's an interesting idea because you'd have no concerns or less concerns about releasing it back into the wild. And obviously, its host food species would be prevalent. Beth, when you mentioned Oriental bittersweet, part of the problem there is we have a Native American bittersweet, even though it's rapidly being swamped out, hybridized with the Oriental bittersweet. But that's the kind of, excuse me, that's the kind of thing where it'd be really hard to find a disease or a insect species that's specific to damaging the Oriental bittersweet, not the American bittersweet. As you kind of saw with that Phragmites research. So it might be interesting. I mean, the verticillium wilt showed up on its own. And I should mention this. I'm looking through my notes right now. I had this written down. And I apologize. Matt Tankos, Tancos, is working down in Maryland, or rather from your perspective, up in Maryland, for the USDA. And I know he is really interested in looking into if someone sees some dying invasives, he's interested in finding out what might be attacking them and culturing that out and seeing if it's something that could be used for biocontrol or kind of pushed in that direction, like you said, of kind of increasing its virulence towards the invasive species. So If anyone sees anything, like I said, Matt Tankos at USDA in Maryland. Let him know.
So Andrew, we do have a live question from Evelyn, and Evelyn, I'm going to allow you to talk. So if you want to unmute yourself and ask your question.
Hello, Andrew. I'm a fellow DEP retiree now, and I have a lot of ailanthus on my property. I'm in Perry County. I have 20 acres, and there is every invasive known to mankind on this property. And I wondered if I could become a part of that Penn State research that is being done somehow, and who I could contact, or if you would know how to get involved.
Rod might actually know a little bit better about getting involved with the research effort. I know that my connections were with that effort were with West Virginia University, Matt Casson, I believe.
Okay.
And at Penn State, it was Don Davis. Is he retired now?
I don't know. I've not heard that he was retired, so I don't know if he's still there or not, but he was the point person for a lot of this.
Yeah. So I would try to reach out to them and see if they have any knowledge about that. We work with some of those researchers. I was working with WVU's Kristen Wickert for a while before she moved on to West Virginia State government, not another. And but yeah, I don't have a direct connection there, and we don't, we're not spreading it. We're not supposed to be spreading it purposely, as I tell our foresters.
Right, yeah, I understand. What was his name again?
Matt Casson at West Virginia University, and Don Davis at Penn State.
Don Davis.
Yep.
Okay, thank you very much. And I like your background there. Is that a window, or is that actually a photograph?
No, that's actually a photograph. I work from home, and my wife runs a daycare. So every other room in our house gets taken over by children at least several times a day. So I'm in my bedroom, which makes my wife very happy when I'm up earlier than her, typing loudly, very loudly on the keyboard, apparently. But so I have my messy bedroom in the background. This is actually a picture of Deer Valley Lake from southwestern PA at our Mount Davis tract.
Yeah, look, very, very nice. Yes, very pretty.
I wish this was my view out of my window.
Well, I actually do have some nice views here of the mountains, and I'm very happy here, and with all the wildlife around me. It being a wildlife biologist in a former life, this is just perfect for me. So
I can't complain too much about my view out my window, but it's no view from the highest point in Pennsylvania.
yes.
Well, thank you, Evelyn, for your question. Appreciate that.
Hey,
And
yeah, Andrew, your laser dot cursor, where'd you get that? That's really cool.
Oh, that's just when you go into present slideshow.
Oh, it's in the Zoom. It's in the Zoom system.
No. This is, I'm sorry. This is when you're in PowerPoint, Windows PowerPoint. Actually, I can show you real quick. I apologize, everyone. This is a little off topic.
I'll look for it. I just
maybe you can show us when we're kind of doing
right here. Hopefully, you can see it. Oh, so hang on 1 second. I'm sorry. I'm already there. It's right here. When you have this view up, it's this right here. So hopefully you can see that. It's an option there under the PowerPoint
when you're in
view.
Okay, great. Thank you. Thanks.
So Andrew, we did have another question from Norma, and they asked if there's anything to be wary of in terms of a landowner releasing biocontrols. So
no, I mean, I would work with any of your state officials to make sure that the permitted in your state, if you're importing, or if you're working with like Philip Lampi, or like I said, at least one of those places out west that I've worked with in the past, all have their permits up to date. These things have been so well studied by the time they're approved for release that I don't think you have to be too concerned about any negative impacts.
Great. Well, thank you, Andrew, so much for joining us this afternoon.
You're welcome. Appreciate it. Hopefully this helped everyone, and thank you for your time.
Yes, well, thank you. And so I'll go ahead and wrap up today. I will go ahead and share my screen with you. So I just wanted to, a few closing remarks. I just wanted to remind you again to register for our upcoming workshops in May and June. So May 26th is our first workshop. And also, as you leave today's meeting, there will be a brief exit survey. So if you can take a few minutes just to let us know how we did and how we can do better and make our content relevant to you. So your experiences and questions help make our programs better. And then lastly, I would like to thank Kelsey and Andrew for joining us this afternoon and sharing their presentations with us, and everyone on the PRISM team who helps to make our quarterly meetings happen. And then thanks to each of you in the audience for spending your afternoon with us and for supporting Blue Ridge PRISM. So with that, we'll close the meeting and have a wonderful afternoon. Thank you.